All right, everybody. I want to spend a bit of time talking about um, how you get from DNA to protein. So how does the nucleus, the control center of the cell that contains the DNA, how does that guide the production of proteins, which do pretty much everything in our cells? All right, so the nucleus is pretty large. It's one of the larger structures in the cell, and you can see that it's surrounded by a structure called the nuclear envelope. It has pores in this envelope, and you can see these pores lead out into the cytoplasm of the cell. Some of them are directly kind of linked to the rough ER. So the visible DNA that we can see inside the cell, this is called chromatin. And there is also a dark stained area called the nucleolus. And this is usually where ribosomal RNA is being generated to make ribosomes. Okay, so the nucleus is the control center for the cell. It contains DNA, all those nucleotides, so the A's, T's, C's, and G's. That code is strung together to code for different proteins. So the information is actually in the sequence of those nucleotides. So the order of those nucleotides on a strand of DNA. So basically, what your cell does, most of the programming in your body is controlled by the sequence of 4 letters. And that determines your cell structure and function. Most cells only have one nucleus. Some muscle cells are multinucleate. Some blood cells all have many nuclei too. These are cells that make platelets. And then red blood cells have no nucleus. They eject the nucleus and they get rid of it because they're big bags of hemoglobin, oxygen carrying protein. So they just don't have space enough for a nucleus. Okay, so we said in the nucleus we find our DNA. And in our DNA, so right here we can see a little picture of the sequence of nucleotides. So this is that sequence that determines, it's basically the code that determines what kinds of proteins you make. And you can see, as we've talked about before, that DNA is a double helix. There's 2 strands of complementary bases. And this double helix in the nucleus is wound around proteins called histones. And when a cell is getting ready to divide, this structure condenses. It packs tighter and tighter and tighter into these supercoils. And that's when we get visible chromosomes during mitosis. During the rest of interphase, these chromosomes are less visible. The DNA is not condensed, and it's harder to see in the cell nucleus. Okay, so how do we get from DNA to protein? We go through 2 major processes called transcription and translation. The first is transcription. So when you transcribe something, you are usually, so if I was making a transcription of this lecture, I'd be writing it down. Or making a copy of that lecture in my words. So going from speech to words. And so in this case, transcription is going from the same language to the same language, pretty much. So we're going from DNA to RNA. So these are two nucleotides, so they're really similar in structure. So in DNA transcription, a gene or the instructions for a protein is copied into our complementary RNA bases. And so we make what's called a message RNA. And that message RNA then leaves the nucleus and it undergoes a process called translation. Now, translation happens at the ribosome. So where are some places we find ribosomes in the cell? If you said in your mind the rough ER or the cytoplasm, you'd be right. So translation happens in a couple different places, the rough ER and the cytoplasm. A lot of the translation that happens in the rough ER are proteins that are going to be packaged into vesicles transported from the Golgi complex or Golgi apparatus into vesicles to the cell membrane or outside of the cell. And then translation that happens on the ribosomes in the cytoplasm are just usually general cellular proteins. So we call it translation because in translation we're kind of changing complete different languages. We're going from a nucleotide language to making proteins. So we're turning this sequence of nucleotides, RNA nucleotides, into an amino acid sequence. And amino acids are what make up proteins when they're all linked together. Okay. So I'm going to step you through these different steps in the next few slides, but take a look at the different steps of transcription and translation. You can always go back and refer to this slide later. Okay, so in transcription, we've got our DNA, and the DNA is unwound slightly, and you've got an enzyme called RNA polymerase. An RNA polymerase comes along and takes one of these strands of DNA and makes a complementary RNA copy. So you can see instead of a T here, you have a U, because that's RNA has uracil instead of thymine. And so you get a complementary copy. So if there's a T here, there's going to be an A here. If there's a G here, there's going to be a C here. If there's an A here, there's going to be a U here, and so on down. So this complementary copy is what is going to be translated into a protein. The DNA, for the most part, doesn't leave the nucleus very much. Okay, I don't want to spend a lot of time on RNA processing in this class. All you need to know about RNA processing is that the RNA that is the complementary copy that's copied off the DNA is sometimes processed. It's sometimes cut and spliced together. I don't want you to worry about the details too much about RNA processing. It's a little above and beyond what we need to go into for AMP1. So after the complementary RNA molecule is synthesized, after it's processed, it moves from the nucleus to a ribosome and then gets translated. So this nucleotide language gets translated into a protein or amino acid language. And so what happens is the ribosome, which we remember, protein synthesis happens at the ribosome assembles around the RNA molecule, the message RNA molecule. And when a ribosome is reading the RNA code, it reads it in triplets, so 3 bases, and that's called a codon. So each of these triplets, depending on where your reading frame is a codon. And each codon codes for a specific amino acid in sequence. So what happens is the ribosome will assemble. It will slide down the RNA molecule and basically read the different codons, and then a complementary transfer RNA molecule will, carrying what's called an anticodon or complementary sequence to that codon, will assemble within the ribosome. And that transfer RNAs, they're called transfer RNAs because they transfer amino acids to the ribosome and to the growing protein. So the transfer RNA is going to come in, and it's going to bind to the complementary sequence on the mRNA, and it's going to bring this amino acid to the site. The ribosome is going to shift down another 3 nucleotides and read this codon next. And then this transfer RNA will bring the correct amino acid to the enzymatic site where they are joined together in a chemical reaction to form the polypeptide chain, to form the growing protein. So you can see the ribosome just keeps moving down the RNA, and it bumps off the old tRNA and keeps bringing in new transfer RNA molecules, and they keep bringing in the correct amino acids to grow the polypeptide chain. And there is a enzyme site here that facilitates a chemical reaction to link those amino acids together as they come in. So that's how we move from DNA, which is a nucleotide, to mRNA, which is a complementary RNA copy of the DNA gene. And then the mRNA is read at the ribosome, and then the transfer RNA brings in the correct amino acids to create the growing protein chain. So this picture is just kind of showing you how we go from code to code. So we've got DNA, and DNA is double-stranded. So we would open up these double strands, and a complementary copy would be made of this template strand right here, a complementary RNA copy. And that's what you get right here. And then RNA is read, this triplet code codes for different amino acids. And when you link together, you don't need to memorize the different amino acids at all. You just need to know that we have about 20 different amino acids, and they get linked together in different ways to make a huge variety of proteins. And so amino acids are linked together into proteins at the ribosome.